And we have a group of over 250 physicians. We have adult and pediatric physicians, hematologists, rheumatologists, and oncologists, and other specialists as part of our group, and pathologists. We also have basic scientists, and we have clinical scientists who provide translational research. And our aim is to advance the knowledge and to improve outcomes for all patients with histocytic disorders under one umbrella. And we coordinate research protocols and provide people to work together. All of you as participants are muted. Please put in any comments you have via the chat feature. If you have a question, put it in the chat. At the end of all the webinar, we will then respond to your questions. If you go to the next slide, please, what you will find is that there might be questions that other people have asked. Just put a like there so that we know that there may be more than one person who wants to ask the same question. We may not be able to answer all questions, but we'll try to do so along the way or at the end of the webinar. Next slide, please. Most importantly, if you're not already a member, do become a member, because it's a wonderful association. It's a family, and you get to learn a lot about histocytic disorders and meet really interesting people. So I'm now going to take you through the presentations. Dr. Milan Minkoff from Vienna is going to talk about LCH-IV, which was the last LCH protocol. The HLH Registry would be with Kai. So I'm now going to step down.
Can you see my screen?
Yeah, we can. Yeah, it's come up.
It's come up. Okay. I think we have very short time, and good afternoon from Vienna. And thank you for this opportunity to update on the LCH-IV study. I have some very exciting news that we can close this study after almost 13 years of ongoing study. And I am presenting here on behalf of the very, very large study management group, which includes the Stratum chairs and the national PIs. Just to remind you that the study was first opened in Vienna in December 2012. It is really close to 13 years, very long time. And I think maybe this is the last study of this type which has been done by the Histiocyte Society due to new regulations and difficulties. Just a brief reminder, the study was quite ambitious. This project aimed to give the best available treatment for any of the different clinical scenarios, and in the same time for some of the patients to answer some scientific questions, and therefore had some strata with randomizations like Stratum 1, Stratum 1, Group 1 and 2, Stratum 2, and the other strata will non-randomized or non-interventional. And it was really unique global collaborations with 25 participating countries on 5 continents. So due to this collaboration, we recruited almost 2,500 patients in total. Here you see some distributions, particularly to Group 1 and Group 2 of Stratum 1. Just to remind you, Group 1 is multi-system disease. Group 2 were the patients with single system multifocal bone disease or CNS risk lesions. Stratum 6 were the patients with singular bone lesions who just were observed to see what is their clinical course and to see how many of them secondarily need systemic treatment. And Stratum 5 was the patients with CNS involvement, either neurodegeneration or tumoral type, and the other strata were mostly secondary, coming from one of those strata. To come to Stratum 1, Group 1 multi-system disease. For the multi-system disease, we had 2x2 factorial design, and we asked the question for those patients who were adding a third medicament like 6MP, or if we continue, we prolonged treatment duration to 24 months will improve the event-free survival, which in the LCH-III trial was about 70%, 60 to 70%. And for this group, we recruited more than 600 patients. Some of them were ineligible for non-response or other reasons. So out of them, 383 have been randomized, and you see that it's quite even distribution. And we wanted to have 400 patients, but due to the fact that we have results which show, now it is for the first time I can show this here openly, that one of those four arms, particularly the standard arm, 12 months treatment duration without 6MP was inferior to the others, and the other had quite comparable results. This means 12 months duration with 6MP was equivalent to those who have 24 months with or without 6MP. I am sure that here will be many questions which I can't answer at the moment. We have the answer we asked, and to see event-free survival. But the reasons why it is this way, is this due to the drug, is this due to the duration or something else? We need some post hoc analysis, and this has not been done. So I think the responses will come at latest when we have the final evaluation and the public data publication. So the recommendations of the DSMC is restricted to those patients who already have been enrolled into the study but are underway, because for the group A, we cannot recommend this arm, it is inferior. So for those, it is recommended by the data set we're talking about to 12 months, to 24 months instead of 12 months. For the 24 months trial, it is enough to have the 12 months with 6MP. So as suggested here, and for newly diagnosed patients, actually for patients who are treated outside of treatment protocols, we have to issue also some recommendations, but at that moment, without complete evaluation, we still cannot do this. We try to do this as soon as possible, because in the past, the standard treatment was 12 months with 2 drugs, and this is the inferior arm here. Therefore, there are 2 options. One is to have 12 months with 6MP and 24 months with or without 6MP have the equivalent results. And I think for the families, maybe the shorter duration will be the preferred option. But at the moment, we still don't have official recommendations for patients outside of clinical trials. The stratum 1 group 2, it was the patients with multifocal bone or special risk disease. We had some baseline from the LCH-III trial, which was not randomized, and we asked ourselves at that time whether prolongation from 6 to 12 months could improve event-free survival as well. And we have recruited more than 700 patients, 442 were eligible and were also randomized. They're quite evenly distributed, and you see that consistent with the data of the Japanese group, which has used different backbone, but has the same conclusion, that prolongation, the treatment, does not improve event-free survival. So here the DSMC had not new recommendations, because we have stopped almost a year ago. But we used the time till now to complete the data. So we have very good data completeness at the moment. There's still a little bit data to be collected, and the publication is in progress. And I think until the beginning of the next year, we can have the publication done. The stratum 2 was a treatment for patients who had either non-sufficient response or patients who have relapsed, but in non-risk organs, or they have patients with response in risk organs, but patients who do not fulfill the criteria to come into stratum 3 to be treated with very intensive treatment. So this group was quite heterogeneous, but the reason was that if you split the subgroups, we will never have good numbers. And here the question was first to use the backbone from the Dutch protocol, and it is a very similar one to the Japanese group. But then the main question was the value of the continuation treatment in those non-risk patients, and to see whether endometriosis, which has been speculated for years, they have few uncontrolled groups treated, whether it is equivalent or it is inferior to 6-MP methotrexate, which has been once again studied with or with other drugs in many historical trials. And we have also here 230 patients, but the design here was for 100 patients. We have more than 100 patients randomized, and once again, you can here see that endometriosis is clearly inferior to 6-MP MTX. So the stratum, the recommendation here is that we have also to close this stratum. Has already been closed, and now we just try to improve the follow-up. And I think the DMC is also decided to can disclose the data, so I present the data now. So the other stratum were non-randomized. The stratum 3 was for the patients with very, very severe disease, and we saw that due to the new the options, due to the inhibitors, availability of inhibitors, that the recruitment in stagnating. Stratum 4, the stem cell transplants have no included patients at all, and stratum 5, we have already 33 patients. If somebody of you is very careful and compares the second page, I show the total recruitment, you'll see discrepancies. This is only because in stratum 5, we allowed the patients who were not on stratum 1 or 2, but developed neurodegeneration to be included and be treated according to the concept here. So therefore, the 24 patients on the second page are coming from the initial strata, and there will be some patients coming from a start treated in all protocols. Altogether 33, because some of the patients have either neurodegeneration and tumoral lesions. There are some combinations. And the stratum 6 was the natural history trial. So the comments is that as those strata are not randomized, once we are now able to close the randomized strata, to close all strata, to close the study, and just keep it open for the patients who are still on the way, already recruited and randomized, until they end up their treatment, have a sufficient follow-up. There have been no safety issues, so this was more or less what I can share to you in this short time. I have to acknowledge the invaluable work of the Data Safety Monitoring Committee. All of those colleagues are already retired, but they give us their time and their expertise. Thank you so much for this, and thank you to all of you and all countries, all clinical sites who have contributed to the study and made it success. I am happy that we can conclude this trial now, and we'll see how we proceed in the future. Thank you.
Thank you. Should we move on to Kai then to talk about the HLH registry? Is he next?
Santa, did you want to ask a question? Like there was one question in the chat, or did you want to leave those to the end? I'm sorry.
I think, excuse me, maybe you can do it now because I have lecture, I have to leave. I have students waiting for me.
Fine. I think there was just one about the results about CNS neurodegeneration, which I think you mentioned a little
We
bit
haven't
about.
evaluated. We haven't evaluated the non-randomized strata. We'll do this shortly because now we know have to do all evaluations, collect all the data, and conclude, and number of publication will follow.
Okay, thank you. Thank you, Milen. Can I invite Kai to start talking about the HLH registry, please?
Yes, thank you, Vasanta. So I hope there aren't any fields on the screen right now, so everyone can see all the slides. So I'll briefly give an update on the HLH registry. Right. So this is the time of the registry. As you may know, the registry was set up in 2016 as a joint effort of the Histiocyte Society and the European Society of Immunodeficiencies. There were some modifications during the years. Last year in 2024, the first results of the registry per se were published in Blood. The registry is recruiting, and so far 324 patients have been recruited. So if you are watching from an ACID country, from the European Society of Immunodeficiencies, you may know that the ACID registry, to which this registry is linked as well, has been put on another platform. So we do data collection on two different platforms with identical data fields. And the ACID platform has been changed, and we have already programmed the HLH registry for this new platform. So if you join from ACID, use the data entry tool of the new ACID registry for the HLH registry. So these are the countries, and as you can see, recruitment is mainly in Central and Eastern Europe and the Middle East, and recruitment is going on in all these countries that you can see. So I only very briefly will mention the publication that was done last year in Blood. As last year on this occasion, I've already given more details on the publication. So this is the distribution of genetic defects, which is quite normal. There's a slight preponderance of FHL3. And one of the key outcome figures was that the etoposide-based treatment, if compared to the HLH 2004 papers, has improved substantially, both pre-stem cell transplant and post-stem cell transplant. So I will now go on to projects that are attached to the registry. So as always, a registry can be a backbone to other projects that can arise from it, with some data from the registry in cooperation with other teams. And I assume many of you have contributed to the FHL2 and 3 study. That is a study that was mainly done by Stefan Eller and his team, which was performed as we expect that genetic newborn screening in the future will pose substantial difficulties how to deal with potential identified genetic defects in terms of what we can offer to the patients. And that will be most relevant in the most frequent genetic defects, which is FHL2 and 3. And the potential future gene therapy trials on FHL2 and 3, which make the natural course and the course after transplant in patients with FHL2 and 3 most relevant. So this is why patients with these two conditions have been collected from the registry, but in cooperation with other groups that do not enter their data into the registry. And so far, about 300 patients have been recruited. Recruitment is closed, and the data collection is now closed as well, and data analysis is continuing. Another project is the A91V perforin project plus X. So as you all know, the variant A91V is quite prevalent in the heterozygous and even in the homozygous variant. It does usually not cause HLH per se, but if it is in trans with another pathogenic mutation, it may cause HLH. But the paper showed quite nicely that this is tricky. So in the upper figure, you can see the perforin expression on flow cytometry, and on the left-hand side, the healthy controls. And then the next plots show heterozygous and homozygous A91V patients. And the red plot shows you patients with A91V plus another substantially pathogenic mutation, and you can see that expression is quite reduced. However, these are results from patients who presented with HLH, and as you can see below, that there were a number of patients that were substantially affected by HLH, 52 individuals. 39 had FHA-related manifestations, 4 had recurrent disease, 15 needed transplant, and 14 even died. So it is an issue. However, there are many other examples in which these constellations do not lead to HLH. For example, in family members that were checked, and where out of 14, only 1 was symptomatic. And in the UK biobank, there were several individuals who had the same constellations and did not have HLH up to the 7th or 8th decade of life. So this is published and can be written in the literature, read in the literature. So we have other projects that are not directly from the registry, but are attached with the registry. As you know, the registry focuses on hereditary forms of HLH. So I assume many of you deal with these patients that have HLH in patients with EBV-infected T and NK cells. Well, I assume the participants from Far East Asia are much more familiar with this than we are. However, so you know that even in patients of European ancestry, this may occur, and we are currently collecting a cohort of these group of patients with severe HLH in patients from Central Europe with EBV-infected T and NK cells, ranging from chronic active EBV infection to overt malignancy. And another project is a project of pathway defect accumulation, as has been published previously. Several heterozygous mutations in different genes or related to HLH, for example, in different degranulation genes such as FHL3 or FHL5 or Griscelli syndrome. So has been accused of causing HLH, even if they're only heterozygous but in different genes. And Stefan L's team is currently working on this with population-based data, if this is truly the case. So these are projects that are set up on the backbone of the registry, well, we hope to give you more information on this in future webinars. So thank you for everyone who has contributed to the registry. It is still open for recruitment, and I'm happy to take questions now or later.
All right, should I go ahead with the next then? So while I'm getting this set up, just thanks to Vicenta for hosting this and Kathy for organizing. It was nice to see so many of our colleagues who are on this webinar in Seattle a couple weeks ago. Hope everyone enjoyed their time in the US and got home safely. So just give me 1 second here. Pull up this. So share my screen. Can you see my screen? Is it my whole desktop or just the you seeing my whole desktop or just the presentation?
It's your desktop.
Okay, let me try again. Sorry. Better? Okay.
Yep, that's it.
Perfect. All right, thanks so much. So I'm Carl Allen. I'm the co-director of the North American Consortium for Histiocytosis, along with Carlos Rodriguez Galindo. And on behalf of our NACHO team and network, happy to share with you current activity and some planned activity. So one of the major activities has been the LCH-IV trial, which of course is now closed. So thanks, Milan, for sharing those data. Just a little bit about the NACHO network. We started back in 2014, 2015 with a foundational grant from St. Baldrick's. And we started off with 11 sites. Our goal was to create a group with a mission to cure all patients with histiocytosis through innovative and collaborative research. And so the plan for this was to create a network to facilitate clinical, translational, and basic research. Basically to create a clinical trials network and also create correlative biology opportunities to better understand the diseases. You can see in the last decade, I think the unmet need is really demonstrated by our kind of unplanned growth from 11 sites to 68 sites now in the US, and we have sick kids in Canada. And we've also been discussing possibility of joining some groups in Central and South America. So NACHO Bio is one of our major studies, and this is an umbrella protocol that serves as a vehicle to collect both tissue as well as clinical data. So the idea was to kind of use this as a universal tool so we could collect things either prospectively, ad hoc, or as part of correlative biology with clinical trials. So in a lot of ways, it was designed in the same manner as the Children's Oncology Group Project Every Child umbrella protocol. So again, this gives us, for consenting patients, it gives us permission to prospectively or retrospectively analyze clinical data and biological specimens. We open enrollment to anyone with known or suspected histiocytosis of any kind, as well as family members. The coordinating center is here at Texas Children's Baylor College of Medicine. So rather than making this sort of a registry or biobank per se, we did build in a few specific aims just to help us power it and design it in a way that was a little easier to manage with IRB. So just some general descriptive biology to identify genes, proteins, molecular pathways that are involved in different types of histiocytic disorders, and then secondary aims, again, to kind of look at biomarkers of risk in correlative biology. And we have used this for some studies and some ongoing studies looking at the clinical significance of different mutations, cell of origin, looking at peripheral blood for risk stratification, looking at plasma profiles to assess tumor burden, et cetera. And this is the basic outline. So when people consent to this study, there's no penalty for not collecting according to a certain protocol, but this is the recommended protocol. And we certainly encourage getting at least one blood sample so we can at least get germline DNA from patients. But the basic idea is that we're going to get a sample pre-therapy after the first couple cycles of therapy and at the completion of therapy, 6 months after therapy, and at any recurrence or change. And so within the protocol, we have this sort of outline for LCH-III, HLH-94, for other all the other NACHO trials I'll talk about in a second. And there's basically this universal approach where, again, we'll take any tissue that's available, but only clinically indicated procedures will take marrow and CSF, pre-therapy samples. This is an error here, but basically after the first couple cycles of therapy, completion of therapy, and then if there's any change or recurrence. So how's this working? I think we've spent a lot of time getting the study sort of set up. To be fair, we don't have a lot of funding for this study, so sites are doing this in a voluntary fashion. We have a little over 20 of the 68 sites enrolled. So we're hoping that as we get more funding, we'll be able to expand this more broadly. And then also to kind of create a little bit more focus as we have specific projects and we do specific correlative biology on clinical trials. But you can see we have a reasonable number of samples that span the histiocytosis framework, especially LCH. Another thing we're working on doing is virtually connecting some of the larger sites. So St. Jude's here at Texas, other sites. We have a lot of institutionally banked samples, so we hope to be able to link those so that both internally and externally collaborators can check the inventory and see if there's opportunities to get samples. Okay, I'm going to switch now to NACHO-COBE. So NACHO-COBE is a phase 2. I'm sorry, I think I missed a slide on this, but it's a phase 2 trial to test the safety and efficacy of cobimetinib in children and adults with LCH and related histiocytic disorders. So in the group 1, this is going to be children with relapsed and refractory LCH. Group 2 is going to be patients of any age with progressive LCH or neurodegeneration. I'm sorry, they have to have progressive neurodegenerative LCH without systemic disease. This can also be, neurodegenerative ECD as well. Group 3 is going to be children with non-LCH disorders, so JXG, Erdheim-Chester, histiocytic sarcoma, et cetera. And then group 4 is going to be adults. So this is sort of modeled after like Eli's study. So the primary objective is to determine the overall response rates in each group. Secondary objective, progression-free survival. And then we have some exploratory objectives to look at the implications of peripheral BRAF V600E, et cetera. And so the correlative biology of this is linked through NACHO Bio. Again, we're looking at peripheral blood mutations and burden of disease, some plasma studies, and some CSF biomarker studies. So this is the basic model. So this is a little bit, I think, different than maybe the model in Europe, where we're basically giving 12 cycles and then we're stopping. And this is sort of intentional. I think we understand that most patients are going to relapse, but we want to see like what really is the rate and kinetics of relapse, understanding that we can resume therapy and treat patients subsequently. And so we have patients on study for 5 years. So in addition to getting this sort of initial response rates, we'll be able to figure out, what happens subsequently, at least, even though it's not dictated by the protocol, we're able to follow those patients and see what their outcomes are. So we give 12 cycles or 12 months of this approach. I should note too that we're giving 60 milligrams per dose. We decrease that in adults to 40 because of the high rates of toxicity in that group. And so where are we now? We actually have enrolled 57 of the 86 planned patients. Group 4 is closed because we've met accrual. Group 3 is closed. So again, this is the adult all-comer group. This is the pediatric non-LCH group. And then we still are enrolling for the pediatric LCH and the pediatric neurodegeneration. So the question is really kind of what's coming next. I think we've talked about this a little bit at the society meeting, but I think we find ourselves in this place where we've learned a lot. We need the biology is coming fast. We have all these different agents we want to test, and how do we do this without taking a decade to run every trial? And so what we propose is kind of using this model where we treat for 12 months with whatever the agent is, and then observe for 12 months. And then when people relapse after COBE, can we try something else? And if they relapse after that, can we try something else? And there's all these different things we could try, HDAC inhibitors, antigen targeting, base treatment medics, checkpoint inhibitors, other MAP kinase inhibitors, chemotherapy, et cetera. I'll say our first study that we have in the NACHO Next series that hopefully will be coming very soon. Kevin Blumwitz is leading this effort, is combining cobimetinib with clofarabine. So it'll be the exact same protocol as NACHO Cobi. It's just we'll set the clock again and start with 12 cycles of cobimetinib and clofarabine. So hopefully this will be open in the, or at least coming out for approval in the next month or two. So here's the team. A lot of people thinking about how we can make the biggest impact. This was actually a project funded by a NIH grant to get stakeholders, including patient advocates, together. So I think hearing from patients about what their experience is increasingly important. Try to incorporate PROs into all of our studies is part of the plan. And I just have to highlight Sarah has really kind of kept us all together. So thanks so much, Sarah Hastings, for being the hub of NACHO. And so this actually is NACHO. Carlos named his dog Nacho when he was a little puppy. He was born with a consortium. Now you can see he's a big dog. He has his own library, and he gives bones out to walkers by. So if you have questions about this, please feel free to send me any queries about the study. And also the NACHO site is listed there. So I'm going to go ahead and stop sharing on this one and tee up Mike's talk. And then as questions come in, certainly happy to answer any. Oops. Sorry for my lack of Zoom skills today. Okay, getting ready to share Mike's screen.
Thank you, Carl. Mike, I presume you're safe somewhere.
Yes, I'm in transit, but I'm not actually driving myself.
Okay, so you continue to share.
So,
Mike, I think you're all teed up.
Okay, great. All right, awesome. Thank you, Carl, and thank you, Vasanta, for the interest. So I want to talk a bit about the Into HLH Registry, where we are partnering with NACHO, and it's active in North America. So I think it's a very good fit here. So we have a website. Welcome anybody to come take a look at it. So, but next slide. The question, of course, is that you've heard from Kai about the Society and ESID joint registry already. And so, why another registry, right? And of course, there are even other registries we haven't heard about today. But the compelling reason for this registry is slightly different, and I think and hope actually will be quite complementary and good for collaboration with the society trial as well, is that there is a lot of detailed natural history questions that we really don't know the answers to, because these sorts of things were never described in the Histiocyte Society studies, or outside of single institution series and so forth. So there are, but these are mostly complicated questions, so it really requires, I'd say, a special methodology. And these gaps are even more obvious for non-familial forms of HLH. So next slide. So we've developed the Into HLH Registry, and so we're running it in a couple of ways. One is that it's largely a patient-powered registry, meaning that we have a website. Patients can actually go visit that website and can electronically consent to participate. With patients' consent, we, instead of the traditional model of sending a form out to a site that's participating, instead what we do is we gather the records centrally with patients' cooperation and abstract them centrally in a detailed fashion. Now, we of course can enroll patients in the traditional way by simply approaching them with a consent document, but the underlying process is the same. That is a sort of a centralized, detailed abstraction. Now, the registry is based out of Cincinnati Children's. It's supported by SOBI, but there are a few limitations, and one is that because we are going through records ourselves, these records have to be in English, and for practical sort of IRB and ethical committee rules, we're really only equipped to acquire records from USA and Canada. And so next slide. And so this is where we are in terms of progress as of last month. So we have 192 patients that have consented to join the registry. But there's a little bit of drop off as this process is not as straightforward as simply enrolling somebody and sending in a form with their data. So there are a number of patients where we can't really obtain their records for practical reasons. So as you can see, the 192 drops down to 160. And then there's the process of actually acquiring the records and prepping them and then abstracting them. And so there's quite a bit in process there. So we have records from 84 patients that have now been completely abstracted. And because we're following patients for up to 5 years after diagnosis, there's the majority of these patients still have additional follow-up data to be obtained. So next slide. And if you were in Seattle, you may have seen a poster with a bit of data that we are generating from this registry. And we are much younger than the Society Registry, so I think there'll be lots to come here shortly. But next slide, you can see a little better, just to highlight a couple of tidbits of those data. So we thought we'd start by looking at this question of diagnosis, which is a really thorny question. Not what the diagnostic criteria should be, but are in the real world, are patients getting diagnosed in a prompt fashion? And so you can look at in panel A, you can see, so you can see the definition, all the way to the left. So we note when the patient initially actually come from medical care. We first and then we have what we call HLH Registry diagnosis, which is
Mike, sorry to interrupt.
Yeah.
But you seem to be going into a poor reception zone. I don't know if anyone else noticed that because it's very interrupted.
I think he just dropped off. I can
Yeah.
finish. I can finish his slides.
Okay. Will you finish takeover? Thank you.
Sure. Yeah, no problem. Yeah, so I think the point is just that this is really a kind of a unique model of getting super granular data. Mike's created this this team and framework to really extract as much data as possible from these patients. And one thing I didn't mention, I neglected to mention in my talk is that with NACHO Bio, we include permission to share the patient's contact information with the IntoHLH team. So in that way, when patients sign up for NACHO Bio, we give them a universal ID. The studies aren't linked formally other than permission for Cincinnati to contact the subjects, and the subjects can then enroll into HLH. If they do so, we'll be able to connect them through GUIDs or universal IDs, and then we'll have kind of biology specimens that mirror the patient samples. So it's been a huge amount of work that Mike and his team have put into this. I think we're starting to see that we're getting some really interesting data from it, and again, thanking SOBI for their partnership to support this registry, and happy to take any questions on either the NACHO part or the IntoHLH.
So I guess I'm next, right? I'm gonna share my screen.
So Mike, I see you're back. I finished on your behalf.
Thank you, and apologies for the connection issues.
So can you see my screen?
Yes.
Right. So I'm Polizois Makras. Hello from Athens, Greece. I'm an endocrinologist, and thank you for I would like to thank the Histiocyte Society for having me here to show how we can how we can map LCH and other histiocytosis through the LCH model, which was developed as a collaboration between three European reference networks, namely the BOND, the EuroBloodNet, and the ENDO-ERN. So some historical features, everything has started back in 2017, where we had the first European registry for rare endocrine conditions. It was named Eureka, and it was 1 year after where when the EREC was launched and the core registry in 2019. So these are the two registries I'm going to present you. And in 2020, we had another European registry for rare bone conditions. It was called EuroBone, and now the two projects has merged in one registry, the Europe registry. So to briefly describe the two registries, the first registry, namely the EREC, is a very simple one, and there is no consent needed for the patient. It is a monthly electronic survey. It can be customizable by the reporters, and it takes less than 5 minutes to complete. You just complete the numbers of the new patients that you have encountered during the last month. And when you put just the numbers, then you take some registration numbers that can be used in the next registry, which is the core registry, where you need a patient consent, and it takes around 5 minutes for core data elements. And there you can have some longitudinal core outcomes. You can have clinician and patient-reported outcomes, and patients can give their measurements, namely their prompts. And of course, for both registries, there is a data access committee, and the data can provisionally be given to researchers upon request. And there are, as you can see, some other affiliated registries. Until December 2024, we had 113 centers from 32 countries. And the number of patients in EREC was around 56,000, and the majority of them were adults, while around 15,000 were children. In the core registry, we had a lower number of cases, of course. We had 51 centers from 20 countries, and the number of patients in the core registry were around 4,000 cases. And I can tell you that during the last year, we have a great increase in these numbers. However, the report's going to be ready at the end of this year. So how to create an account in the core registry? You can feel free to take a snapshot of this screen, because you can use this link for the self-registration, and any center could become an active participant. And if your center is not listed here, you can add it and fill in the form, choose the core registry clinical contributor role to have access to the core registry. And your request you fill in your data will be approved as soon as possible. And then you can start adding patients. It is quite easy, as you can just click to add patient, and you can have, of course, some test cases to familiarize with the system. And as you can see, there are many diseases that can be included in the core registry. But if we were talking about LCH and other histiocytosis, we have to use the systemic and rheumatological conditions. And then in the diagnosis specific outcomes, we can choose Langerhans cell histiocytosis, either unifocal or single system multifocal, or pulmonary LCH or multisystem LCH. And this here also we can have Langerhans-Chester disease, Rosai-Dorfman, or other non-Langerhans cell histiocytosis. And as I told you, in the core registry, you need the patient consent for the access to this registry. And after ticking the corresponding answer of patient access, this button will appear automatically in the menu, and you need to know your patient's email address, because the system will send the invitation link. And also you can write an additional message and choose the preferred language. There are many languages there. And on the platform, patient can see the information about him or her and fill in the prompts upon a request. Here are the patient's data that should be filled in. As you can see, it is an easy system. You just click, for example, the disease activity, the biopsy specimen, whether the patient had a BRAF testing or other mutations were tested, and the organs involved. And we have a sub-page for the treatment used, lines of treatment in the past, current treatment, if it is known or not, and the type of targeted treatment if we are giving our patients a targeted treatment. And the last page, sub-page is other malignancies and comorbidities, and you can choose whether your patient has any of them. So these are the ways to contact. You can use this link, as I told you before, or you can just email to this email address. The central administration offices are in the Leiden University Medical Center, and you can ask them to how to use this registry. You don't have to be in Europe. You can do it from everywhere in the world. And these are my acknowledgements. I would like to thank the condition-specific module development group. It took us around 10 to 12 months to have this new module. And I hope you will find it interesting and get on board. Thank you very much for your attention.
Thank you, Moses. I don't think there are any questions in there. And that was probably the last of the presentations. Kathy, have you got the... Yeah. Thank you. So could everyone please provide us with a response to the survey? We can only make this better if you tell us what works, what doesn't work, and what you'd like to do differently. If you have any suggestions of things that you would like to see. And remember, this is an update on current projects and studies. This isn't an education update, but do let us know what else you'd like to see in these webinars. Did any of the speakers have any comments? Or there's some questions that have popped up. Olive, I've seen your question. That was probably for Milen, and I will pass it on to him. Daniel, thank you.
I think we have a question from Jan. I can actually allow him to talk, which I didn't know I could do. So I'll let him.
Okay.
Go ahead, Jan. We can't hear him. Did you want to type out your question? We're not able to hear you for some reason.
I've responded to Trung's question. Thank you, Jan.
I think those are.
I think that was more a comment.
Yeah,
Yes.
maybe you can hear me now or not?
Yeah, we can
Yes.
hear you, yes.
Oh, okay. Yeah, I was just saying it's a very easy, the core study is very easy to participate in because it's already there and you only have to put somebody who is the PI in your institute in leads to make to put LCH in also in the registry for the center that you're working in. So it's pretty easy and it's yeah, I would say it's good to participate. Well done, Polisois. That's I think.
Thank you very much, Jan, for confirming that. Be on board.
Thank you, everyone. We shall see you again in a couple of months or 3 months. Thanks to all the speakers. And if you have any other queries, you could reach out via the society or directly with speakers. Kim, was there anything else you wanted to add at all?
Don't forget to fill out the survey for the Histiocyte Society meeting in Seattle, if not already mentioned. So just to make sure that we can further improve upon the meetings next year. So thank you, everybody.
Thank you all. See you soon.
Bye.